Hello, welcome student. We shall continue the last topic, that is classification and nomenclature of enzymes. Classification and nomenclature of enzymes. I have already told you that normally enzymes are classified into one is simple enzyme, and next conjugate enzyme. In the simple enzymes, only protein part is present, which I have already told you. In the conjugate enzymes, protein plus non-protein part is found. Protein plus non-protein part is found. Protein means enzyme is mixed with another non-proteinaceous part. That non-proteinaceous part is called cofactor. It is called cofactor, and that time the protein part or enzyme part, whatever that, it is called apoenzymes. This is called apoenzymes, and the part, non-protein part, which is added to the protein or enzymatic part, that is simple protein. That non-protein part which is attached to the simple protein or proteinaceous part is called cofactor, and the enzymatic part is called apoenzyme. Non-protein part is up to 3 types. One is prostatic group, second one is coenzyme, and third one is metal ion. Understands? These two are organic compounds and this is inorganic. So in that organic compounds, tightly attached with apoenzyme. It is tightly attached with apoenzyme. This is prostatic, coenzymes, and metals, and the cofactor is divided into 3 parts. This is the organic compound. It is tightly associated with the apoenzyme. As for examples, hemoglobin, it is heme plus globin. Globin is a simple protein or protein, that is enzymes, and this is a heme is non-protein compound. So non-protein compound is attached to the globin or protein. So heme is the prostatic group and which is attached to the protein. So it is the example of prostatic. The hemoglobin is example of that. Heme is the prostatic group and globin. Next, the H2O2 by means of enzymes peroxidase change in H2O plus O2. This is also an prostatic group, that is OH2 and O2. Means such type of enzymes which catalyze the compounds into 2 others. So it is the first. Now coenzyme. Coenzyme, it is the also organic compound, but it is loosely attached with apoenzymes. Examples: NAD, that is nicodinamide adenine dinucleotide; FAD, flavin adenine dinucleotides there. Now the metals ions. Metallic ions, it binds the protein and help in adding or removing of electrons. It helps in the adding or removing of electrons. It is the examples. By losing the electrons, it becomes triple ions. So such type of cofactor is metal ion cofactors, and which is binds with the apoenzymes. So these are the certain types of the cofactors, and so simple apoenzyme is bind with the cofactor, then it is called conjugate protein. Now the new latest classifications which is internationally accepted, and that classification is given by IUBMB, International Union of Biochemistry and Molecular Biology. International Union of Biochemistry and Molecular Biology. This is the international unions, and this union gave its classification, and on that basis the enzymes are classified into 7 classes. The enzymes is divided into or classified into 7 classes. Although in your book and in some that 6 classes are mentioned, but latestly means generally one more class is also added to it. So I shall tell you about all the 7 classes which is at present found. So this class 1, 2, 3, 4, 5, 6, 7. There's 7 classes, 1, 2, 3, 4, 5, 6, 7. For your convenience, O, T, H, L, I, L, T. For your convenience, you can say O, T, H, L, I, L, and T. These 6 name classes are given in your book, and that the new class is 7. That. So one is oxidoreductase. Oxidoreductases transferases, hydrolysases, liage, isomerase, ligases, and this one is translocases. These 7 names are given. 7 class. These are the class. And on such basis, on the basis of this class, the nomenclature is mentioned, and that nomenclature is based on 4 digits, means name is based on 4 digits. Understand? Their name is based on 4 digits. Means what the digits means, by their digits we can classify or we can give the name of any enzymes. So this is the most important of. So 4 digits is most important. On that basis the name is given. This 4 digit is called enzyme commission number. Understands? The 4 digit is called. Suppose there is enzymes number 2, 1, 3. Suppose this is the 4 digits of an enzyme. Understand? So what does it means? The first digit means first digit will tell about its class. Means 2 means it is the of the class transferases. If there is 1, that is of oxidation. If there is 5, that is isomerases. So first digit, that is 2. Understand it? It mentions its class. Second digit it mentioned subclass. Second digit it mentioned subclass. Third digit mentioned subclass. Third digit mentioned subclass. And the fourth digit mentioned individual enzyme number. So this means this is the third digit. This will mention the individual enzymes. These subclasses, that is, they are differentiated on the basis of their properties. Means that is the class, and these are the subclasses or subclasses is determined on the basis of their properties, what the properties they have, and that we will study in the upper classes. And this is the individual enzymes numbers. And so it is the, suppose 2, it means transfers. If I say 1, 1, 1, 1, means it is the first member of class 1, that is oxidoreductase, and 1, this is a subclass. So it is a member of oxidoreductase, and it is the example of aldehyde. So in this way the 4 digit express the name of the enzymes. This is the 4 digit principle, and which is called enzyme commission number. Oxidoreductase, where you see that both oxidation and reduction are found. It catalyzes both oxidation, catalyze the oxidation and reaction. Understand? And one thing you know, that oxidation and reductions occur simultaneously. Understand? Means always though वो साइमल्टेनियस यानी ऑक्सीडेशन और रिडक्शन दोनों साथ साथ होते हैं. अब यानी अब S1 plus S2. Here you say if here is X1 S2 this one. Now say what? Understand? As for example, a loosely examples you see that Na plus Cl NaCl, understands? Plus. Now you see that Na gives electron, so it donates electrons, so it oxidizes, and it takes electrons, means oxidation. Na gives Na plus ion and Cl minus ion, means here is oxidation, here is reductions. Oxidations means that is lose of electron, and reduction means gain of electrons there. And now it oxidize, it reduce, means here is oxidation, here is reduction, so it is just opposite. One is oxidized, that is what reduced, and that is reduction, that is oxidized. So we can say that here first reduction, S1 reduced and S2 oxidized, and that opposite S1 oxidized and S2 reduced. Just reverse the case, understand? One that is go on oxidation, in the next it will be reduced, and one which go in the reduced form, that will be going in oxidized form in the next one. Now the oxidoreductase, also known as dehydrogenase. Example is alcohol dehydrogenases. Understand? That is reduced, is oxidized, it is oxidized, that is reduced. Means oxidation and reductions always occur simultaneously, एक ही साथ दोनों होते हैं. So एक ऑक्सीडाइज होगा तो दूसरा रिड्यूस होगा, और जब ये रिड्यूस होगा तो वो ऑक्सीडाइज्ड होगा. नाउ ट्रांसफर ऑफ ग्रुप्स, मान लो ग्रुप्स भी है, हाइड्रोजन के अलावा अदर्स देन हाइड्रोजन कुछ भी हो सकता है, बिटवीन ए पेयर ऑफ सबस्टेट एस और एस डैश के बीच में ये ग्रुप है जो आगे this transfer of groups from this to that, means glucose when add with ATP, understand? So it becomes glucose 6-phosphate, understand? So means this ATP is added to that, means that goes on its 6th position, and here is enzyme hexokinase, it is the transferase. Now the third one in hydrolysis. Hydro means water, addition of water. By adding water, it's breaking the covalent bonds. Examples, when maltose is hydrolyzed with H2O, it will give glucose plus glucose. Similarly, sucrose on hydrolysis with H2O, it will, by the enzyme sucrase gives glucose and fructose. So with the help of H2O, the bonds between covalent bonds between them is breakdown. Now lysis, non-hydrolytic removal of functional group, that is removal of functional group from that. This one is from substrate. Examples, pyruvic acid. The on removal of CO2 change into acetaldehyde. And so this is the isomerase, change the aldo group to keto group and vice versa. I mean both, from that aldo to keto and keto to algo. Examples, THAP, dihydroxyacetone phosphate. In the respiration you will get change into 3-phosphoglyceraldehyde. It is forms 3-phosphoglyceraldehyde. That is, it is a keto, it is aldo. Understand? So enzymes is isomerase. So here is interchange of aldo to keto. Aldo means CHO is the functional group of aldehyde, and C double bond O is the functional group of keto. So aldo to keto is changed place by means of enzymes isomerase. It catalyzes the linking together of two compounds. These are two, that is ligase, that is catalyzes CS bond PO like that. As for example, pyruvic acid plus CO2 TP, it changes into oxaloacetic acid by means of enzymes carboxylase. Translocase, it also the transfer of ion across the plasma membrane. Understand, that is membrane. So this takes place by the help of enzymes ATPase, and that is the enzymes takes translocase, that is ATPase is a type of translocase enzyme. So these are the 7 classes of the enzymes. That's all.